I'd like to thank the organizers of the conference as well as Aubrey for inviting me today. What I'm going to talk about is some early studies in terms of looking at tissue engineering of the thymus. And what we're trying to do is to produce a thymic tissue ex vivo that will have function in vivo. So our goal for this project is to engineer a functional thymus ex vivo, and to do that, to meet that goal, we've got a couple of objectives in terms of developing decellularization protocols to produce thymus scaffolds. So we think the natural scaffold will offer a lot of advantages in terms of engineering this particular organ. And we'll also characterizing the scaffold in terms of its extracellular matrix content as well as some of the growth factor and cytokine retention on that scaffold, and that's why we think that will be important in terms of regenerating this organ. So the rationale is the thymus undergoes involution during aging. T-cell immunity decreases with age, and as you saw yesterday from a very nice talk, the reduction in T-cell repertoire or the reduction in naive T-cells during the aging process, and particularly under certain circumstances, you can get a shift toward increased presence of T memory cells as you age. So what's the significance of engineering thymic tissue? One of them is to reversal of age-related changes in the thymus. We also may be able to use these constructs to enhance immune recovery following immune suppression, particularly in cancer treatments. Potential treatments for autoimmune disease, you may be able to reset the immune response in people that have autoreactive T cells. And also tolerance induction and allograft transplantation. You may be able to induce central tolerance and get rid of immunosuppression in these transplant patients. So just a little bit background on the thymus. It develops from endoderm that's derived from the third pharyngeal pouch during embryological development. One of the important transcriptional factors is FOXN1. Without that, you get no thymic epithelial seeding of the organ, and the phenotype is the nude mouse. It's an athymic animal. You also get some neural crest mesenchyme cells present within the developing thymic organ. These provide some of the support growth factors for thymic epithelial cells. Lymphoid progenitors from the bone marrow seed the thymus very early in embryological development, and there is evidence that you also get additional lymphoid precursor seeding that occurs in waves during adult life. So this is a picture of the thymus. It contains a cortical region and a medullary region, and there are specific cell populations of epithelial cells in these two regions. There are cortical epithelial cells and medullary epithelial cells, and they have different functions. Other cells that are very important in terms of education of the T cells in the thymus are dendritic cells, and I said some of the fibroblasts provide some of the growth factors. This is a schematic diagram of some of the early developmental stages of thymocytes as they develop, mature in the thymus, and it's been fairly well characterized. And there are different stages here denoted by DN1, 2, 3, 4. This stands for double negative, and if you look at these thymocytes, you have CD4 or CD8, and they don't express those, and that's why it's termed double negative. And then you come into a double positive, so it expresses both CD4, CD8, and then final maturation of a single positive. It chooses between the CD4 expression or CD8. CD4 gives you helper T cells, CD8 positive cells, or cytotoxic T cells. And you can look at other surface markers. Expression of certain cytokines are very important during this developmental stage. as well as gene rearrangements for the T cell receptor. There's a very specific and unique pathway for these developing thymocytes. They come in from the bone marrow precursor cells, come into the corticomedullary junction area. They start their maturation process, and they start a migratory pathway from the corticomedullary junction toward the capsular region within the cortex. They undergo their maturation process. During this part, a very important selection process of positive selection occurs. They then mature to single positives, move into the medullary region, and undergo negative selection, hopefully to eliminate any self-reactive T cells, and then they exit the thymus as mature cells to go to the periphery. Now, as I said, within the thymus, there's cortical epithelial cells. Those are responsible for positive selection of thymocytes. There have been a couple of molecules here, the serine protease and a proteasome subunit, that have activity in this positive selection of specific CD4 and CD8 populations. And the medullary epithelial cells help in the negative selection of thymocytes, as well as some dendritic cells are very important in the negative selection. And all of this is associated with an autoimmune regulator, transcriptional factor called AIR. And in the mouse, a common progenitor epithelial cell has been identified, particularly in the embryological developmental stage. There's been some difficulty identifying this in the adult animal. As we age, the thymus involutes or decreases in size. There have been attempts on the clinical side to reverse some of that degradation of the thymus, and one of those is you can inhibit some of the sex steroids by using the luteinizing hormone releasing hormone agonist, and that reverses some of the thymic involution. Growth factors such as keratinocyte growth factor has been used. It increases some of the cellularity within these aged mice. And clinically, it has been used in chemotherapeutic patients to reduce some of the mucositis. A cytokine that's very important is interleukin-7. In some cancer patients, IL-7 expands the naive T cells and increases the overall T cell repertoire. But again, these are all transient. Once you stop treating, the effects go away within the thymus. There have been a number of ex vivo T cell production assays developed many years ago in terms of using both thymic stromal layers that contain the epithelial cells, some agar cultures and thymic organ cultures. These do produce T cells, but the whole process is very inefficient. There was a group of about 3 publications that occurred in the early to mid 2000s using a solid matrix material. It's a carbon-based matrix, and they seeded them with thymic stromal cells and bone marrow progenitor cells, and they could get T cell production out of this 3D or 3-dimensional culture system. And very interesting, there was one publication that used human fibroblast as well as skin keratinocytes from humans, and they found that this was able to produce T cell maturation. But there's some controversy in terms of using keratinocytes. They express some of the similar markers as thymic epithelial cells, but this is an ectodermally derived epithelial cell. Thymic epithelial cells are endodermally derived. So we wanted to approach this in terms of using a scaffold, but we chose to use a natural scaffold through a decellularization process. And this is a standard decellularization process using a detergent, in this case Triton-X. You can use a number of other detergents. And we do extensive washing. And to get rid of the released DNA from the lyse cells, we use a DNase treatment. I'll show you a little bit of data that may or may not be the best thing to do. and then more washing, and then we're looking at different assays to characterize these scaffolds. So this shows an HE stain of a native thymus tissue, both mouse and pig, medullary region, cortical region. This is after decellularization, and you've got the matrix material that's the only thing left. If you look at the DNA after 2 days of decellularization, you can see significant reductions in the amount of DNA. But if you use DNase, you can almost eliminate any of the DNA present on that scaffold material. However, if you look at this is native tissue. This is freeze-dried material. This is after decellularization. And then if you look after DNase treatment, you can see significant degradation of that scaffold. So we're concerned in terms of what's happening during this process. This also can be seen in scanning EMs. This is the decellularization, picture of the decellularization of the scaffold, and then after DNase treatment. And you can see appears to be significant damage to the scaffold after this treatment. We wanted to characterize what molecules were present on the scaffold. This is native tissue stained with a Masson's trichrome, which stains collagen blue. In the mouse, you can see there's still a little blue remaining in the scaffold. In the pig, there's much more dense collagen remaining in the scaffold. And using Alcian Blue stain, looking at glycosaminoglycans, very little is remaining in the scaffold. So we have some problem in terms of this whole procedure eliminating some of the ECM components. Now we also further characterized these scaffolds looking at collagen, specifically collagen 4. This is a native thymus tissue, and this is after decellularization, and you can still see some of the collagen 4 present. Laminin in the native tissue, and you still see some present in the decellularization, following decellularization of the scaffold. Now with fibronectin, it's a little bit different story. We have significant amounts of fibronectin in the native thymus, but it's almost completely eliminated during the decellularization process. So this is one of the molecules that appears to get washed away or eliminated during this whole process. So one of the things we wanted to look at then is how effective will these scaffolds be in terms of enhancing proliferation of epithelial cell populations, maintaining their phenotype in culture. So what we did was we looked at the decell thymus scaffold from a porcine model, as well as taking whole thymus tissue that we freeze-dried and cryomilled and made a powder. And we did the same thing with the scaffold. We cryomilled it and made it a powder. And we also looked at the mouse whole thymus as a cryomilled powder. And if you add that to two-dimensional cultures of epithelial cells, and you plate the epithelial cells after 1 week, the epithelial cells do undergo proliferation, but in the presence of the scaffold or cryomilled material from the thymus, we see an enhanced proliferative response. And after 2 weeks, the decell thymus scaffold actually gave us the best results. We're not sure why we didn't continually see proliferation after 2 weeks with the whole thymus from pig or from mouse. It may also contain some inhibitory activity for proliferation. We have to actually look at this a little bit closer. So we then grew epithelial cells and looked at some of the phenotype changes that occur during culture. If you take thymic epithelial cells, place them in culture, they lose their expression of EpCAM, as you can see here. This is a 1-week culture of thymic epithelial cells, and they don't express EpCAM. But if you look at whole mouse thymus, you can see that there's abundance of thymic epithelial cells expressing EpCAM. After 1 week of treatment with the powder from the porcine native thymus, we get very nice upregulation of EpCAM in these cells. If we use the pig decellularized scaffold, we see a little bit, but it's not as pronounced as using the whole tissue cryo-milled powder. Cytokeratin 8 is one of the cytokeratins that is expressed in the cortical region of the thymus. You can see that very nicely expressed here. This is the medullary region with very little staining of cytokeratin 8. If you look at thymic epithelial cells, they don't express cytokeratin 8 when you place them in culture. However, after exposure to the native thymus cryo-milled material, you get very nice upregulation of cytokeratin 8 within these cells. You see a little bit with the decell scaffold. So we may, what we're seeing here is probably some of the elimination of some of the factors during the decellularization process. Now, if we look at cytokeratin 5, which is expressed in the medullary epithelial cells, the cortical regions are up here, less expression, more expression in the medullary region. You do see expression in thymic epithelial cells that have been cultured for a week, but if you add this powder from the native thymus, you get much more upregulation of expression of this particular cytokeratin, and you also see some with the pig decell scaffold. So what we're seeing is this material has the ability to upregulate the proliferation of the thymic epithelial cells. It also induces and maintains the appropriate expression of these thymic epithelial markers, EpCAM and the cytokeratins 8 and 5. We also looked at the potential upregulation of some of the important transcriptional factors, in this case FOXN1. We can see this is native thymus. It decreases if you just place epithelial cells in culture. And then in the presence, we can see a small increase with the mouse native thymus powder. We expect to see a much greater increase once these epithelial cells have direct contact with bone marrow progenitors or thymus progenitor cells. There is a two-way communication between the epithelial cells and the thymocytes to help mature both of these populations. Data that we actually just got this week in terms of taking these scaffolds, and we actually used the cryo-milled material and acid extracted what we could get off of that material, and then looked at a cytokine array as well as a growth factor array. And what we see out of the cytokine array is that we can find some IL-1 present there, a little bit of GM-CSF. In some samples, it's much higher than others. We see IL-12, as well as some chemokines. And these chemokines are very unique in terms of being more specific for T cells and not macrophages, monocytes. So IL-16, this is a monocyte inducible interferon gamma chemokine, as well as MIP-1 beta. So all of these are involved in bringing in T cell populations. So that may be extremely important in terms of recellularization of this scaffold. If you look at the growth factor analysis, we have FGF, which is important in terms of looking at epithelial maintenance and growth. We have insulin-like growth factor, as well as some insulin-like growth factor binding proteins that are present in these scaffolds. I was a little disappointed in the data. I was expecting to particularly see maybe some IL-7 and some IL-4, other important cytokines, which we didn't see, which may be eliminated again during the production of the scaffolds, or actually how we acid extracted these to get the cytokine arrays. So actually, in conclusion, we can take thymus tissue. It can be decellularized using standard detergent methods. The use of DNase to remove the residual DNA further damages the scaffold. So we're actually looking at this very closely. How important is it to remove the residual DNA once you lyse the cells? We're not sure. We're going to move to a little bit milder conditions in terms of decellularization process and see if we can maintain a little more of the growth factors and cytokines. Although there was some damage to the scaffolds, they exhibited functional properties such as maintaining some of the growth factors and cytokines. And these scaffolds also had the ability to induce the proliferation of thymic epithelial cells, as well as maintain the epithelial phenotype in culture. And all of this was done in 2D culture. We're now moving into three-dimensional culture using hydrogels, and as well as taking these 3D cultures and going in vivo, particularly right now in a nude mouse model to see if we can get T-cell production. I'd like to thank a number of people that have been involved directly in periphery with this project from WFIRM, as well as the SENS Foundation for support of this project. And I'd be happy to take any questions.